Hello, dear students. So, our topic for today is all about carbohydrates. So, this is the second topic for your midterm period. Now, as you can see, this will be our objectives. So, di ko na po isa-isahin pagbasa. You will find that in your handout. So, I do hope that your handouts are ready because as we go along our lecture, you will be able to find that in the handout and para ma-follow nyo rin yung ating discussion. For the topic outline, we will be discussing about the general description of your carbohydrates, what are its classification, its metabolism, and lastly, the clinical correlations. Of course, for the laboratory methods or the laboratory side of carbohydrates, that will be discussed in your laboratory subject. So let us start with the general description. So, carbohydrates are the most abundant organic molecules in nature, in which these are the different functions of your carbohydrates. Carbohydrates are our major energy source in the form of your glucose. Carbohydrates can also be stored uh, to be used as energy for later in the form of your glycogen. Carbohydrates are also components of our cell membranes, especially when we talk about your glycoproteins, and it could also be a structural component in plants, bacteria, and insects. Now, from when we try to look at the structure of your carbohydrates, it actually contains your carbon, your hydrogen, and your oxygen molecules. In that, your carbohydrates contain a carbon double bond O and an OH functional group. So, bale, looking at the molecules present in the structure of your carbohydrates, what differentiates carbohydrates, especially to your protein, is that your protein contains your nitrogen. Now, these three images that you see here are actually the different structures or different projections that show uh, the different carbohydrate na structure, as mentioned. So, there are three. The first picture here, this first image that you see here is actually what we call your Fisher projection. Now, I guess this is already just a recap on your side since mm, the different projections discussed here are also discussed during your biochemistry. So when we say Fisher projection, this shows a linear structure. The second image here is a cyclic na structure, in which this is what we call your Haworth projection. And then for the last image here, this is what we call your chair conformation. So it is called as such because you can see para daw siyang chair, or it is also possible to relate it as a boat conformation. So these are the three structures of your carbohydrates in which it can be presented in this ways, and the importance of showing these different structures, you will get to appreciate that later on when we reach your classification. So let's proceed to the classification. Now, there are four different classification properties of carbohydrates, in which the first one is we try to look at the number of your sugar units. Second is to look at the size of your base carbon chain. Third is in terms of the location of your carbon oxygen functional group. And lastly, in terms of your stereochemistry. Now, again, we will not really dive deeper into this discussion since, as mentioned, but medyo na discuss na rin ito during your biochemistry. So, what we are going to do is just to recap and then yung general description lang din for each. So, as you can see here in this table, this is already the overall. This already presents the overall classification of your carbohydrates from the number of sugar units to your functional group. Nakapresent na po dito lahat. But then again, we'll go over it one by one. Start po tayo with the number of sugar units. So when we say number of sugar units, we are referring to the monosaccharides, your disaccharides, your oligosaccharides, and your polysaccharides. Now, monosaccharides here, kids, They are also called as your simple sugars. They are your simplest form of sugar, and they are actually your basic unit of carbohydrates. When we say basic unit, meaning to say the rest of this here, your disaccharides, your oligosaccharides, and your polysaccharides, they are composed of these basic units. Kumbaga, kung sasabihin natin na this is an oligosaccharides, meaning it is a chain of monosaccharides forming a certain compound or substance. Now, examples of your monosaccharides are your glucose, fructose, and your galactose. Next, your disaccharides. Examples would be your maltose, your lactose, and your sucrose. Oligosaccharides. These are chains of 2 to 10 sugar units. While for polysaccharides, these are more than 10 sugar units. An example for polysaccharides are your starch, glycogen, and your cellulose. So going back to disaccharides, when we say disaccharides, meaning to say dalawa, two monosaccharides being combined together, disaccharides are formed by condensation reaction, as you can see here, condensation reaction between two monosaccharides. And the linking of these two monosaccharides, the bond that is present there is what we call your glycosidic bond. So as you can see here, our three main disaccharides are your sucrose, your lactose, and your maltose, in which, as you can see here, dito po nakasulat kung ano po yung basic unit or monosaccharide na composition nitong mga disaccharides. So for your sucrose, we have your glucose and your fructose, a combination of your glucose and fructose. For lactose, that is your galactose and glucose. For your maltose, we need two glucose units. And of course, once these disaccharides, not just your disaccharides, but all, including your oligosaccharides, including your polysaccharides, once they are hydrolyzed, nakukuha natin yung mga monosaccharides na. Hydrolysis would result in getting our monosaccharides. So for oligosaccharides, since it is not mentioned here, these are examples of your oligosaccharides. Again, oligosaccharides are chaining of 2 to 10 sugar units, and the common oligosaccharides that we have is what we have your raffinose, we have your stachyose, hope I pronounce it right, and then we also have your verbascose. So these are just some examples of your oligosaccharides. Now we proceed with your polysaccharides. Again, polysaccharides more than 10 sugar units, and there are actually two types of your polysaccharide. So you have your homopolysaccharide, and we also have this heteropolysaccharides. So when we say homopolysaccharides, kids, it contains only a single type of monosaccharides. And these examples, your starch, your glycogen, and your cellulose, these are actually examples of a homopolysaccharide in which this starch, glycogen, and cellulose all are composed of your glucose. Kumbaga, if it is composed of, for example, kunwari si starch composed of 20 monosaccharides, lahat yung monosaccharides na yun, puro yung glucose being condensed together to form your starch. While your heteropolysaccharides, from the word hetero, these are complex carbohydrates which are formed by combining your carbohydrates with non-carbohydrates or any derivative of your carbohydrate. And an example of your heteropolysaccharide would be your pectin, your chitin, and your heparin. So again, if a layman's term man nato ang pag-describe kay heteropolysaccharides, dili siya purely of one kind na monosaccharide. Combination na gyud siya, combination of a non-carbohydrate, a derivative of your carbohydrate, and such. So, here in our starch, glycogen, and cellulose, one thing that could differentiate the three is that your starch and glycogen are composed of an alpha glucose, while your cellulose is made up of a beta glucose. Now, as to what is this alpha and beta, We will discuss that further later on when we reach your stereochemistry. So, tandaan nyo po muna yan. So, let us further differentiate these polysaccharides. So, syempre na-differentiate na po natin the cellulose na again, its subunit is that it is composed of your beta-glucose. So, how about your starch and your glycogen? Now, starch, there are actually two types of starch. You have your amylose and you have your amylopectin. Now, one thing that differentiates amylose and amylopectin is in terms of its structure, wherein your amylose is just a linear molecule, while amylopectin is branching as you can see here. So, ang problem na lang po natin is differentiating amylopectin with your glycogen. Kasi nga 'di ba they are the same subunit, alpha glucose, same din po sila na branching. Well, in terms of branching, if you look at the structure of your glycogen, mas highly branching pa si glycogen. And if you try to look at the branches, how these branches are made, dito din po sila nag-differ. In which the branching of your amylopectin is per 20 subunits, while the branching of your glycogen is per 10 subunits. So yan po yung different types or examples of your polysaccharides. So now we proceed to the second classification, which is the size of your base carbon chain. So, bale, from the name itself, all you need to do is to count the number of carbon present in its chemical formula or structure. And thus we have these terms triose, tetroses, pentoses, and your hexoses. So, from the word tri sa triose, meaning there are 3 carbon chain present, tetros, 4, pentos, 5, hexoses, 6. And the monosaccharides that we have mentioned a while ago, your glucose, ito po, the monosaccharides that we have mentioned, your glucose, your galactose, and your fructose, these are examples of your hexoses. So how about your triose, your pentose, and your tetrose? Now, triose sugar, kids, we have your glyceraldehyde and your dihydroxyacetone. And the smallest carbohydrate is actually your triose sugars, specifically your glyceraldehyde, including din po si dihydroxyacetone since it also have 3 carbon in its structure. How about tetroses? An example of your tetrose, kasi wala po dito sa ating image, so let me just write this one. Example of your tetrose would be your erythrose, your threose, and your erythrulose. This is letter U, erythrulose. So again, these 3 are examples of your tetroses having 4 carbon in its structure. While for pentoses, kids, ito po yung pentose. So you have your ribose or your ribulose. So ganyan po yung ating classification based on the size of your carbon chain. Now we proceed with the location of your carbon oxygen functional group. Now, carbohydrates can be further differentiated by the functional group that is present in their structure. So we have your aldose and we have your ketose. Now sa aldose, the functional group that is present is your aldehyde, while in ketose, the functional group that is present is your ketone. Now look at our image here at the bottom, which represents kung ano po yung itsura ng ating functional group. So recall that there are a lot of functional groups in our body and that was also discussed in your biochemistry. But here, let us just focus on your ketone and your aldehyde. Now, halos the same si ketone and aldehyde and that they both contain a carbon double bond O, carbon double bond oxygen. What differentiates them is the number of R group that is present. Since here in our ketone, may dalawang R group po tayo. Here in aldehyde, isa lang. Yung isa is hydrogen na. So what is this R group? R group is just an abbreviation for any group in which a carbon or hydrogen atom is attached to the rest of your molecule. So meaning to say, pwede po tayo may ididikit dyan na other molecules pa. So that what the yan po, that is what differentiates your ketone and your aldehyde. So here for aldose, the carbonyl carbon is at the end, while your ketos, its carbonyl carbon is at any other position, or I would rather say that it is actually in the middle. So going back to our image here, other than grouping your carbohydrates in terms of the number of the carbon chain, the size of the carbon chain, it can also be grouped according to the functional group. So that is why, kung mapansin nyo, even if they have the same number of carbon present, since may different the functional group sila, so its name also differs. Now, why I mentioned that the carbonyl carbon of your ketone is in the middle, because pansinin nyo po itong structures dito. Now, why is it found in the middle? Because take note, going back to the R group, di ba yung R group is at both ends the carbon. So that is why it is found in the middle, because at both end the carbon, pwede tayong maglalagay pa ng iba pang molecule. Unlike here in your aldehyde functional group, since permanent si carbon double bond O and your hydrogen, kaya nakikita sila sa terminal end. Kasi isang atom or molecule lang yung pwede natin na idadagdag dun sa ating functional group. And that is why we have here your, for your triose, if your glyceraldehyde is composed of your aldehyde group, kaya nga from the name, di ba, glyceraldehyde, while here in your ketone group, we have your dihydroxyacetone. So, ganyan lang po for the location of your carbon-oxygen functional group. Now we proceed with the stereochemistry. So we have here four, your isomers, your epimers, your enantiomers, and your anomers. Now when we say stereochemistry, we are just actually looking at the spatial arrangement of your atoms. Or basically, we are just looking at the arrangement. And then from there, we are able to add certain name, may idadagdag lang sa naming sa ating compound. So let us start with isomers. Now, isomers are compounds that have the same chemical formula. Now, look at your glucose, your galactose, and your fructose. Even if medyo nag-differ sila in terms of positioning of some molecules, they are actually isomers with each other. And when we say isomers, again, same ang chemical formula. Bakit? Ano po ba yung chemical formula ni glucose, galactose, and yung fructose? Glucose, galactose, and fructose all have 6 carbons, 12 hydrogens, and 6 oxygens. And diba, remember, glucose, galactose, and fructose are under your hexose sugars because, again, may 6 sa kanilang carbon chain. And that's what makes them isomers with each other because they have the same chemical formula, C6H12O6. So, what about epimers? Epimers are isomers that differ in configuration around only one specific carbon atom, but with the exception of the carbonyl carbon. When we say carbonyl carbon, kids, or your carbonyl group, this is your functional group that composed of a carbon atom with a double bond of your oxygen. So, going back to your glucose, we have here galactose and mannose, in which glucose and galactose are epimers at your carbon 4, whereas glucose and mannose are epimers at carbon 2. So, same po ang chemical formula, yes, C6H12O6 pa rin tayo. But try to find a certain or specific area in the structure in which they differ. And that is what we call epimer, referring to which portion in the structure they differ, even if they have the same chemical formula. So, kaya sinabi kanina na epimer at carbon 4. Why? Look at the position of your glucose in terms of the OH group. Yung OH ni glucose sa fourth carbon niya is nasa right side, while for your galactose, nasa left side. How about your mannose and glucose? Same, C6H12O6, pero sa nag-differ, they are epimer at your second carbon. Why? Look at the position of your OH. OH position ni mannose is on the left side, whereas the OH position of your glucose is on the right side. So, ganun lang din po yung epimers. So, now we proceed with your enantiomers. Now, enantiomers are optical isomers or stereoisomers, meaning to say that these pairs of structure are mirror images of each other. So, try to look at this image here on the right side. But if you look at the D and L glucose here, same chemical formula, differs in the position in which, again, mirror image. Parang binaliktad lang na, di ba? Look at the position of your OH and H here. Kung si D glucose, everything is on the right side. Pagdating kay L glucose, nasa left side. So that is why the D and L na terms here or designation is referring to the position of your OH group. So the enantiomers are designated as D sugar or L sugar. D stands for dextrorotatory, L sugar stands for your levorotatory. Now again, balikan ko po yung sinabi ko, look at the OH group. Ma'am, ang dami pong OH group sa structure. Saan na specific na OH group? Doon tayo sa tinatawag natin na penultimate carbon. So what is this penultimate carbon, kids? When we say penultimate carbon, it means second to the last na carbon. So going back to the structure here, di ba? 1, 2, 3, 4, 5, 6. And since we are trying to find the penultimate carbon, which is second to the last, so we need to look at this fifth carbon. Since siya man ang second to the last. And then try to look at the position of your OH. If your OH group is on the right side, we designate that as letter D or dextrorotatory. If the OH group is on the left side, designate that one as your L or your levorotatory. Very easy to remember, L would stand for left. Siyempre yung naiwan na si D, automatic, right na siya. And most of the sugars that is present in our body, the common one is actually in the D form. So again, just remember D and L for enantiomer. So now we proceed with the last part for our stereochemistry, which is your anomer. Now, when we try to look at anomers, kids, what we are trying to, the structure that we need to look at are yung cyclic nyo na structure or your Haworth projection. Diba? The rest of the stereochemistry, from your isomer to epimer and enantiomer, kay Fisher projection tayo, but this time around in anomer, we need to look at the Haworth projection or your cyclic na structure. So anomer, cyclic monosaccharides or glycosides that are epimers, differing from each other in the configuration. Now, if our compound is an aldose or aldose na classification, we need to look at the first carbon. If ketose siya, we need to look at the second carbon. Now, I will explain this one further regarding C1, C2. Another classification lang din kay anomer is that we would also call this carbohydrate compounds as furanose or pyranose depending on the structure of depending on your cyclic structure. Na if it is a 5-membered na ring, that is what we term furanose. If it is a 6-membered ring, that is what we call your pyranose. So, ganito po yan siya. From your Fisher projection, we transform it into your Haworth projection. And this is what we are referring to pyranose and furanose kills. Again, pyranose if it is a six-membered ring. So of course, this is a six-membered ring. While here in your furanose, this is a five-membered ring. So very easy to identify. Look at the structure, diba? Look at also the position of your oxygen. Now, anomeres can be differentiated, or yung designation for anomere is the alpha and beta na word. So, ito po yung sinasabi natin kanina, dun po sa example natin sa starch. Di ba, what differs starch from your cellulose is in terms of the alpha glucose and beta glucose. So, dito po natin siya madidiscuss. Now, saan po natin titingnan si alpha and beta? For alpha and beta anomeres, Haworth projections represent cyclic sugars as having essentially planar rings with the OH at the anomeric C1. Now, what we are trying to look at is the anomeric carbon here. Iba po yung penultimate carbon, second to the last carbon sa ating Fisher projection. This time around, we are looking at the anomeric carbon. When we say anomeric carbon, it is the carbon derived from the carbonyl carbon of the open chain form of your carbohydrate molecule. And san yung carbonyl carbon natin? But again, that is the carbon that is near to your oxygen or yung may carbon double bond O. So, next to your oxygen would be your carbonyl carbon or your anomeric carbon. Dito po na, and usually it is in the first carbon sa inyuhang sa ating ring structure. So, once we find the anomeric carbon, what we need to look at next is the position of your OH. If the OH here in your anomeric carbon is below the ring, that is alpha. If your OH is above the ring, that is your beta. Thus, naataani ng mga terms na alpha D, glucopyranose, beta D, glucopyranose. So, easy to remember. Just tandaan nyo lang na reverse yung ating yung ating letters. What do I mean by reverse? Diba? B below starts with letter B. So, dapat hindi siya beta, kundi the reverse or the opposite of it. So, that is alpha. Above is letter A. What is opposite of it? So, dapat beta. That is how you are going to remember your alpha and beta anomeres. And I hope that makes it easier for you to remember. Kasi, yan yung aking parang mnemonics na rin. So, we are done with stereochemistry. So, we are done discussing this for. So, just a recap. Again, isomers are molecules with same chemical formula. Epimers, same chemical formula but differs at a certain position sa kanilang configuration. Enantiomers, yung mirror images. And then we have here your dextrorotatory and levorotatory, your DNL. Levorotatory, left side. Dextrorotatory, right side. Anomers, meron po tayo nung pyranose and furanose. Then we have the alpha and beta. Alpha, opposite, below. Beta, opposite, above. So we are done with our classification. Now let us proceed with the metabolism. So here in the metabolism, we will be covering also your hormone regulation. So of course, since we are talking about carbohydrates, since we are talking about glucose being our main source of energy, it is also good to point out or siguro just a refreshing your minds lang din in terms of our digestive function. So, kumbaga, I know you already memorized this one. So, syempre, hindi ko na rin po ito isa-isahin. What I would just like to point out here is that there are a lot of organs involved for us to be able to get an energy supply, and that each of these organs also have different substances being secreted in order for our carbohydrates and other substances pa from the food that we eat to be metabolized, to be utilized. Diba sa mouth pa lang, andun na si amylase that will actually help in digesting polysaccharides. And then from the mouth, dadaan sa pharynx, dadaan kay esophagus, dadaan kay stomach. And then sa stomach, ang dami-dami ring substances dyan. We have your hydrochloric acid, we have your pepsin, and then even si liver, si gallbladder, and pancreas would help also in the metabolism, digestion, and other necessary means for us to get our energy supply. And syempre, after passing there, agi sa large, agi sa small, and then yung mga excess would be, syempre, in the form of stool, i-excrete din out through passing through your rectum and then your anus. So take note also na ang laki-laki din ng role ni small intestine in terms of absorbing our nutrients in the body. So take note, kids, that only monosaccharides are being absorbed in the body. Kaya nga, there is a necessary breakdown and catabolism of your polysaccharides because, again, yung natatanging ina-absorb at ginagamit ng ating body is in the form of your monosaccharides, which is the simplest form of our carbohydrates. And going back to what I have mentioned a while ago, in terms of absorption, what carries out most due sa digestive processes and absorption of our nutrients is the small intestine. Ang laki-laki ng role ni small intestine nito, kids. So that is why it is also important, take note, how this absorption process is going to take place. So as you can see here, in the image sa left side, this is your small intestine, in which in the luminal side or apical side of your small intestine, there we will find your different mga channels, your SGLT1, which is for your glucose and galactose, and we have your GLUT5, which is for your fructose. While on the basolateral side, you will be able to find your GLUT2, which is for all types of your monosaccharides. Now, again, this is in relation to absorption. Channels ni siya in order for your nutrients to pass through. And these are just some specific channels which in turns may specific din po sila na substances that they are able to transport or channel in and out into our uh into our cells. When we say luminal side or apical side, kids, it is referring to the side in which facing your lumen, kaya nga luminal, ito po, luminal side. While when we say basolateral side, it is facing your bloodstream. Na kumbaga, if substances pass through that basolateral side, parang sinasabi na rin po natin na we are actually transferring these nutrients directly into your bloodstream. So, for you to better or to further understand pa, to better understand this concept, meron po akong video na ipe-present. So, try to watch this video. I guess this video is around mga 7-minute long. So, let me just play this vid.
Hi, and welcome to another episode of Wonders of Chemistry with Mickey G. In this episode, I'll be focusing on how the end products of carbohydrate digestion, namely glucose, is transported from the gut and into the bloodstream. I will discuss where this occurs, the cells that are involved, and how specific transport proteins help to facilitate this process. Let's begin with an illustration of the GI tract, and more specifically, the location of the small intestine. Let's now enlarge a section of the small intestine so we can look at the finer detail. You will see that the walls of the small intestine are made up of 4 layers. The serosa, or outer layer, the muscularis, the submucosa, and the mucosa. The innermost layer of the gastrointestinal tract is composed of simple epithelial cells. you will see that this layer is highly folded. These finger-like projections, termed villi, increase the surface area of the lining of the small intestine. Now, an increase in surface area allows digested foods to come into greater contact with the cells responsible for their transport into the body. Okay, let's now have a look at an individual villus. You will see that each individual villus has its own capillary network of blood vessels, highlighted in red and blue, with red signifying the arterial end and blue signifying the venous end. In addition, you'll also see a central yellow finger-like structure. This represents the lacteal, which is responsible for the absorption and transport of fats within lymph vessels. Now, at the top of the villus, I have circled a number of cells that make up the epithelial layer and lining of the small intestine. These cells are known as enterocytes. Here is an illustration of such a cell. On one side of this cell, and more specifically the side that faces the inside of the small intestine, you will notice a brush-like border. This represents the microvilli, which is where absorption of food occurs. Now, the space within the GI tract where food travels and is absorbed is termed the lumen, depicted here. Now, on the opposite side of the brush border is where the capillaries are located. It is the job of these enterocytes to transport the end products of digestion, such as glucose, from the lumen of the GI tract and into the bloodstream. Let's now have a look at how this is achieved by focusing on the internal machinery that is responsible for this process. Once again, here is a labeled illustration of an enterocyte, with the basal lateral surface facing the lumen, while the other side, termed the apical surface, faces the blood capillaries. Now, as you are aware, glucose is the end product of carbohydrate digestion, and more specifically polysaccharides. This glucose collects within the lumen of the small intestine, awaiting transport. Now, this transport process requires three important steps. Step 1 begins on the apical side of the cell and uses primary active transport in the form of a sodium-potassium pump. This process uses ATP to pump sodium out of the cell and potassium in. Over time, the concentration of sodium within the cell becomes low due to the continual removal of sodium. This low intracellular sodium concentration triggers step 2. And more specifically, a sodium-dependent glucose transporter located on the basal lateral surface. Sodium moves along its diffusion gradient from the lumen of the GI tract and into the cytosol of the cell, bringing with it a glucose molecule. This type of transport protein is frequently referred to as a symporter, as it carries two entities together in the same direction. Eventually, the glucose concentrations within the cell begin to rise above that of the surrounding tissues, and more specifically the extracellular fluid and blood capillaries on the apical side. This brings us to the third step, which involves a third type of transport protein termed GLUT2, and works via the process of facilitated diffusion, and is located at the apical surface of the cell facing the blood capillaries. In this final step, glucose is transported out of the cell and into the bloodstream along its diffusion gradient. Once in the bloodstream, glucose is transported to the liver via the hepatic portal vein. So, in summary, the transport of glucose from the lumen of the GI tract and into the bloodstream requires three main steps, each of which requires a different transport protein. During step 1, primary active transport involving a sodium potassium pump sets the scene by creating a low concentration of sodium within the cell. Step 2 involves secondary active transport and more specifically a sodium glucose transporter on the other side. Sodium enters into the cell, bringing with it glucose. Finally, during step 3, glucose exits the cell and into the bloodstream utilizing a GLUT2 transporter and facilitated diffusion. One important thing to know about this whole process is that sodium is crucial to the transport of glucose from the GI tract and into the bloodstream. In fact, this is the reason why many electrolyte drinks have sodium, to enhance this process. Thank you for listening, and don't forget to subscribe to keep up to date with my future presentations.
Ayun. So, pasensya kids. So far from what I have searched sa YouTube rather, sa kanya yung pinaka-best na although medyo yun nga lang, grabe ka malumanay, grabe ka slow pagka-explain, but it was really the best so far sa ako ang pag-search sa YouTube, which really represents in terms of how your uh, different monosaccharides are being absorbed, especially in the small intestine. Okay. So, now we proceed with the pathways in your glucose metabolism. Siyempre, di na po natin babalikan yung step by step nito. You just recall the general, kung ano yung general description and purpose of these pathways. Siyempre, graduate na tayo dun, di ba? Tapos na kayo sa biochemistry. So, just recall its general function. Now, there are a lot of pathways of your glucose which help in terms of maintaining balance of our glucose level in the body. We have your glycolysis, in which it involves metabolism of your glucose molecule to become pyruvate or lactate for the production of energy. You have your gluconeogenesis, which is the formation of your glue-6-phosphate from your non-carbohydrate sources like your lactate, your glycerol, or your amino acid. So always remember the word neo there from the gluconeogenesis. The word neo would refer to getting energy source from non-carbohydrate sources. Then we have your glycogenolysis. Remember the word glycogen and lysis. Diba? When we say lysis, meaning breakdown. So glycogenolysis is the breakdown of your glycogen to become your glucose to be used as energy. And then when we say glycogenesis, remember the word glycogen and also the word genesis. Diba? When we say genesis, parang creation. When we say glycogenesis, it is the conversion of your glucose to glycogen to be stored because glycogen is our storage form of glucose. Then, lipogenesis and lipolysis also involves or also a part of our glucose pathway in which when we say lipogenesis, conversion of your carbohydrates to fatty acids, while lipolysis is the decomposition of your fats. Kids always know the word, always look at certain key terms because kasi yun na nga, know your word. Andun na kasi minsan sa word kung ano yung definition or kung ano yung function nila. So just like what I have mentioned, 'di ba, mga lysis, genesis, glycogenolysis, the word glycogen, gluconeogenesis, the word neo. So from there, nagkakaroon na tayo. We already have the idea as to what it is all about. So, it is also good to point out as to what is this catabolism and how can we obtain energy from this. Going back to the definition of catabolism, when we say catabolism, it is part of our metabolic process in the body in which it breaks down, baga it involves breakdown of large complicated molecules into smaller ones in order for us to produce or to get energy. And catabolism involves three different stages. Stage 1 would involve the breakdown of your complex molecules to their component building blocks. Stage 2 will involve the conversion of these building blocks to acetyl coenzyme A, which will participate into your citric acid cycle. And that is for your stage 3, metabolism of acetyl coenzyme A to carbon dioxide and formation of your ATP, which is our source of energy. So, bale, looking at this image, of course, your protein, your polysaccharides, your lipids, these are all complex structures in which each of these has its own building block or simpler forms. So we all know that amino acids are the building blocks of protein. Kaya upon the breakdown of protein, we would be able to get the amino acid. Siyempre kay polysaccharides, meron tayong monosaccharides. And for your lipids, we have your free or your fatty acids. Now, very easy in terms of the process of your polysaccharides to be converted to acetyl coenzyme A. Just remember your glycolysis. Eh, di ba, even here in glycolysis, meron na po tayong nakukuhang ATP. But the product of your glycolysis will proceed to your citric acid cycle, which is this one. And diba, remember, the product of your citric acid cycle will proceed in your electron transport chain, giving us more ATPs to produce. How about your protein and specifically your amino acids? Amino acids could participate in your citric acid cycle and in the production of energy through transamination, in which, di ba, pyruvate, amino acid can form your pyruvate, your acetyl coenzyme A, and amino acids are also a part of your citric acid cycle. Whereas for your fatty acids, it will undergo beta oxidation para may acetyl coenzyme A na ma-produce, and then in turn, participate kay citric acid cycle and then participate sa electron transport chain, and now we would be able to get our energy source. So, ganun lang po talaga yung stages ng ating catabolism. Breakdown of complex molecule. Stage 2, conversion of your building blocks to acetyl coenzyme A in order to participate to your last two na cycles, your citric acid cycle and electron transport chain. And for your stage 3, dun na po yung formation of our ATP. So now we proceed with hormone regulation. Before pointing out the different hormones involved in maintaining balance for our glucose level, just remember that during a brief fast, glucose is being supplied to the extracellular fluid from the liver. Kumbaga, there will be there will also be conversion of your glycogen to glucose to be used since syempre nag-fasting nga. When we say fasting, wala po tayong food intake. Now, a fasting period longer than a day, that's the time that glucose will now be synthesized from your non-carbohydrate sources. But syempre, wala naman siguro magfa-fasting na longer than a day. Starvation na din yan. So, these are the different hormones involved in regulating our glucose level. We have your insulin, your glucagon, epinephrine, cortisol, growth hormone, ACTH, thyroxine, somatostatin, and incretins. And yes, we will discuss each of these one by one. Pero, brief description lang in terms of what will stimulate it or what is its function, and saan ba siya sinisecrete, and ano yung kanyang action. And these hormones can actually be divided or can actually be grouped according to its action, wherein looking at the levels of your glucose, those hormones involved in increasing our glucose level are the following. So, ito po yung nag-i-increase ng ating glucose level. Yes, marami. Whereas, those involved in lowering our blood glucose level, meron lang po tayong dalawa. But the major hormone involved in lowering our blood glucose level is really your insulin. In terms of incretins, somehow parang masasabi rin natin na indirect. indirectly lowering your blood glucose level, and we'll further discuss that later. One unique hormone which involves both lowering and increasing your glucose level depending on what our body needs is your somatostatin. Either po siya, either it could increase or decrease your glucose level. Then again, we'll discuss that further once we reach that hormone. So, just an overview in terms of our glucose homeostasis, in which our kung major hormone na nagalower kay glucose level is your insulin, the major hormone that really increases your glucose level would be your glucagon. Where, in case na may increase, ito po, once our body recognizes that there is a rising in our blood glucose level, especially after eating a carbohydrate-rich meal, it will give a signal to our pancreas, which is where our insulin is being produced, to release now your insulin. Now, one signal that could actually activate also the release of your insulin if our glucose level will reach more than 120, more than 120 milligrams per deciliter. So once insulin is now released in our bloodstream, dalawa po yung possible action niya. It will signal our cells to take up more glucose. It will also give a signal to our liver to take up glucose and store it as glycogen. Or kumbaga, insulin enhances the glycogenesis, the process. And then that our blood glucose level, by the time our blood glucose level will normalize, it will now give a signal to our brain to stop releasing the insulin. And then of course, na-achieve na natin yung tinatawag natin na balance or homeostasis. How about glucagon? Siyempre, ang magsistimulate sa release ni glucagon if there is increased removal of excess glucose or if there is low blood glucose level, especially if, again, you are undergoing fasting. So what happens there is that our brain will tell our pancreas to release glucagon since that is also where our glucagon is being produced. And what happens here is that glucose will tell liver, kasi nga dun sinustore si glycogen, so glucagon will tell the liver to break down glycogen, convert it back to glucose, and release it into the bloodstream in order to correct the low blood glucose level in our body. And once it normalizes and reaches a balance, magbibigay lang din po ng signal ang ating body sa brain na to stop the production now of your glucagon. And usually, the low, yung level of glucose for us to know na mababa na siya is andito na range. Once it reach less than 80. So again, that is the overall picture of glucose homeostasis. Now, it is also good to point out what pancreas is all about and the different cells that your pancreas contain. Now, one thing that is kumbaga unique of pancreas, or actually most of our organs, have an endocrine and an exocrine function. When we say exocrine function, kids, remember enzymes. Kasi more on enzyme involvement ito eh. Excuse me. Icebreaker. While when we say endocrine, this is more on related to hormonal functions. And thus pancreas have both this exocrine and endocrine function. For the exocrine function of your pancreas, it involves your acynar cells in which it secretes your digestive enzymes, which will help, of course, in the digestion. Whereas for the endocrine function of your hormones, the cell that is responsible there is your islet of Langerhans cells, which in turn secretes your different hormones. And there in the islet of Langerhans, meron pa po tayong different pa or mga specific cells pa. And each of these cells secrete also specific na hormones. So for your alpha cell that is responsible in the secretion of your glucagon, for beta cell kay insulin, for delta, that is somatostatin, for your PP cells, that is your pancreatic polypeptide, and for your epsilon cells, that is your ghrelin. Siyempre, each of these substances have their own roles and functions in our body. Yan lang din yung gusto ko pong i-point out dito. In terms of the enzymes being secreted in the acynar cells, remember your amylase and lipase enzymes. Okay? So, now let us begin. Let us start with insulin. Now, insulin, as mentioned, they are secreted in the beta cells of our islet of Langerhans, and what stimulates the release of your insulin would be hyperglycemia, or if there is increase in the levels of your glucose. What are the actions of your insulin? So, it will promote increased glucocellular entry, especially into the muscles and adipose tissue. Will also increase your glycogenesis, your lipogenesis, and glycolysis processes so as to use up your glucose and to remove glucose into your bloodstream. And insulin will inhibit your glycogenolysis na process. Kasi nga, our aim here is to lower your glucose level. If hindi i-inhibit si glycogenolysis, remember that the action of your glycogenolysis, diba, is again, glycogen, lysis, breakdown of your glycogen to become your glucose. And glycogenolysis increases your glucose level, thus it needs to be inhibited. Now, in terms of the structure of your insulin, initially it is actually synthesized as a precursor polypeptide which is known as your pre-proinsulin. Kumbaga, what we are trying to point out here is that in the body, hindi po insulin agad-agad yung napoproduce. Pre-proinsulin po muna, then magiging proinsulin, saka pa makuha yung insulin mismo. And in terms of getting insulin, pre-proinsulin here, kids, will first need to pass through your endoplasmic reticulum, wherein there will be a cleavage. Kumbaga, tatanggalin ito na portion ng ating pre-proinsulin, which is your signal peptide, leaving now what we call your proinsulin. Proinsulin will pass through your Golgi apparatus, further cleaving off, or kumbaga, kinakat pa, yan may yung proteolysis or cutting off yung ating structure, and thus we will now be able to get insulin. Other than insulin, meron din po tayong tinatawag na si C-peptide. So, the mature insulin molecule and your C-peptide are stored together and co-secreted from your secretory granules in your beta cell. C-peptide is actually an important marker of insulin synthesis and secretion, as stated here. Again, they are co-secreted, meaning to say, every time that we produce insulin, C-peptide is also there. So, C-peptide is an important na marker, kids, meaning to say, there are laboratory tests that would entail for us to measure the level of your C-peptide because marami siyang use. C-peptide could actually could actually differentiate endogenous and exogenous insulin. Kasi syempre, when we say exogenous, meaning to say we are taking insulin uh, outside of the body, meaning to say we are orally taking it, syempre makaka-differentiate siya. Kasi nga, exogenous insulin does not have C-peptide with it. Kasi insulin na nga siya. Whereas the endogenous insulin undergoes those process of proteolysis and always co-secreted with your C-peptide. Another importance of C-peptide is that it could also actually differentiate your type 1 and type 2 diabetes mellitus, wherein in type 1 DM, there will be a low level of your C-peptide and insulin, while in your type 2 diabetes, there is a normal or high level of your C-peptide. Thus, again, C-peptide is important as a marker for us to know these things. And since C-peptide is being cleared more slowly than your insulin, thus it is a useful marker also of your insulin secretion. Why is it cleared more slowly than insulin? take a look at the half-life of your C-peptide here. Half-life of your C-peptide is actually around 35 minutes. Compare it with the half-life of your insulin na around nasa 4 to 5 minutes lang. So, kaya maganda siya na marker kasi mas matanggal siya na magsistay dun sa ating body. And other than that, C-peptide has no biological activity. It is necessary, though it does not have any biological activity, still it is necessary for the correct structure of our insulin. So, yan yung importance ni C-peptide. Now, in terms of the action of your insulin, ganito po siya. In a cell, this is an example of our body cell, there are glucose channel. You can actually find a glucose channel and an insulin receptor in our different cells, in which once your insulin is attached to the insulin receptor, it will open up your glucose channel, thus increasing the uptake of your glucose, increasing the entry of your glucose to the cell, and in turn, lowering your blood glucose level. Parang ganun yung mechanism ni insulin. Sige, we are done with insulin. Now we proceed with your glucagon. And as mentioned, glucagon is your main hormone in order to increase your glucose level. Glucagon is secreted in the alpha cells of our islet of Langerhans in the pancreas. And what stimulates glucose other than the fasting state, it can also be stimulated during stress. And the action of your glucagon would be, siyempre this time around, i-enhance na natin si glycogenolysis, including your gluconeogenesis. But then again, as stated, gluconeogenesis only begins when there is a fasting of longer than 1 day, or if our carbohydrate sources in the body is already depleted. Na kumbaga, ubos na talaga yung carbohydrate natin. Saka pa, magpo-proceed si gluconeogenesis. Once all of our carbohydrate is exhausted, that's the time that we will be using your non-carbohydrate sources na. Kasi nga, wala ng carbohydrate. So, and then, need tayo ng energy. So, saka pa magsa-start yung gluconeogenesis. And then, again, this is the primary function of your glucagon. Next, let us proceed with epinephrine and cortisol. So, bakit ko ginawang as one, the epinephrine and cortisol? Because they are both secreted almost on the same gland. They are both secreted in the adrenal gland, pero nasa different portions sila ng adrenal gland being secreted. Epinephrine is secreted in the adrenal medulla portion of your adrenal gland. Cortisol is dun sa adrenal cortex, specifically the zona fasciculata. Both are stimulated during stress. Kaya nga, kids, ito ha, di ba pansin nyo, pabalik-balik yung stress. Kaya nga, when we are stressed, there is really an increased chances of gaining weight. Kasi nga, grabe yung pag-stimulate or ang pag-stimulate and release kay glucose sa bloodstream natin. So, for the action, almost similar lang din naman ng action si epinephrine and cortisol. For epinephrine, it will increase our blood glucose level. It will inhibit insulin secretion. It will increase glycogenolysis and it will promote lipolysis. How about cortisol? Cortisol on the stimulation of your ACTH, your ad adrenocorticotropic hormone. It will increase your blood glucose level. It will decrease the entry of your glucose into the cell, and it will also enhance your gluconeogenesis, your glycogenolysis, and your lipolysis. Ganun po, almost the same. So, I would just like to emphasize further on the location of your epinephrine and cortisol. Again, they are both secreted in the adrenal gland. And remember that your adrenal gland is composed of two parts. You have your medulla and you have your cortex. Now, sa cortex, si cortex divided pa into three different zones. You have your zona glomerulosa, zona fasciculata, and si zona reticularis. Each of these zones may specific na hormones na sinisecrete. For zona glomerulosa, dun si aldosterone. For zona reticularis, dun yung ating androgens, yung mga testosterone natin. And for cortisol, ito po yung zona fasciculata. Zona fasciculata is located at the middle portion of our cortex. So, ito, cortisol. While adrenal medulla is the innermost portion of your adrenal gland, and dyan po natin nakukuha si epinephrine, including your norepinephrine. Di ba, these are your fight and flight hormones. So, now let us proceed with growth hormone and ACTH. So, bakit ko din po ito kinumbine? It's because they are both secreted in your anterior pituitary gland. What stimulates the release of your growth hormone are the following: sleep, stress, exercise, and low glucose level. Though growth hormone is not really that valuable as we grow older, pinaka-grabe or pinaka-evident yung function niya during the growth years sa bata. While for ACTH, its stimulus is when there is a decreased cortisol level. Kumbaga, ACTH indirectly increases our glucose level because it controls the release of your cortisol. So once there is a decrease in your cortisol level, ACTH will be stimulated and thus giving a signal na need to release more cortisol, and then cortisol will in turn kasali na yung action niya na yung na-mention natin kanina. So for growth hormone, increase blood glucose level, increase glycolysis, decrease entry of your glucose. For ACTH, increase glycogenolysis and gluconeogenesis, and then stimulate cortisol release, thus increasing your plasma glucose. So, let us proceed with thyroxine. Thyroxine is secreted in the thyroid gland, specifically in the follicular cells, and its stimulus is in terms of the control of your TCH. TCH means your thyroid stimulating hormone. So these are the actions of your thyroxine: increase glycogenolysis, increase gluconeogenesis, and it also increases your intestinal absorption of glucose. So, ito po yung ating thyroid gland. Inside the thyroid gland, ganito po yung makikita natin. San po yung follicular cells? This one. Yung parang nag-encircle ng fluid portion inside. And if you try to look at it under the microscope, ganito po yung itsura niya under the microscope. Ito pong parang fluid-like inside, this is what we call your colloid. What encircles your colloid is your follicular cells, and that is where your thyroxine is being produced. Yung colloid dyan sa center, that is somehow where they are being stored. After thyroxine is being secreted or produced, isi-store sila dito sa colloid nyo. Dyan sila nakatambay until such time that your TSH will be released and stimulate its release. So next, last two for our hormone regulation, we have your somatostatin and your incretins. Now, for your somatostatin, it is secreted sa delta cells ng ating islet Langerhans. Other than sa pancreas, kids, it is also being secreted in the duodenum, specifically on the GI cells of your duodenum. GI cells is like the D cells of your or delta cells of your islet Langerhans. Now, remember what I have mentioned, the somatostatin is actually just in between. Pwede siya maka-increase, pwede siya maka-decrease. Why? Look at the action of your somatostatin. It is an inhibitory hormone from the word the statin, from the word statin, referring to or related to inhibition na siya. It is an inhibitory hormone. Ano yung i-inhibit niya? Si insulin and glucagon. Other than insulin and glucagon, it will also inhibit your growth hormone and other endocrine hormones pa. So that is why it is in between, because it depends on what is needed in the body to maintain balance. Na if sobra-sobra na si insulin, siya yung mag-i-inhibit. Sobra-sobra na si glucagon, siya rin yung mag-inhibit. Kaya indirectly, it could either lower or increase your glucose level. How about your incretins? Incretins are gut hormones secreted by your enteroendocrine cells. It is secreted minutes after eating, and these are examples of your incretins. These are specific names of your incretins. So you have your glucose-dependent insulinotropic peptide, and you also have your glucagon-like peptide. Doon sa dining incretins, what is the role of your incretin? Remember that incretin is under the lowering of your blood glucose level. And if you could still recall, di ba na-mention ko po na indirectly lowering your glucose level. Why? Because the action of your incretin is actually to stimulate your insulin release. Thus, going back to the role of insulin, insulin lowers your blood glucose level. Kaya siya indirect kasi it does not directly lower your blood glucose level. What it does is to stimulate your insulin, in which si insulin yung maglo-lower. And other action of your incretin also is to inhibit your glucagon release. Thus, again, lowering your blood glucose level. Incretin, we could actually say, or other action ni incretin is, it is an appetite reducer. Oh, it is an appetite reducer, and incretins could actually promote weight loss. I am not sure, pero may nabasa po ako about incretins na sometimes it is what is being given to those who wanted to lose weight. Si incretins kasi nga, yun nga, appetite reducer kasi siya. Okay, so that ends our discussion for the hormone regulation. So, di ba, natapos din natin lahat-lahat, and then those are just an overview as to the actions of these hormones. The deeper discussion of these hormones will be, doon na natin siya sa Clinchem 2 nyo, ipapa sa ubaya. Okay? So, for the last portion of our discussion here in our carbohydrates regarding clinical correlations, kindly proceed to the part 2 of our video. So, cut po muna natin para may short break din, and baka relax-relax din kayo. Baka nag-information overload na rin. So, see you on our next video.